Okay, so we're diving into something pretty critical today, a major issue hitting our oceans hard.
Yeah, definitely.
It's this disease, stony coral tissue loss disease,
Mm-hmm.
SCTLD for short.
Mm-hmm.
It's just been tearing through Florida's coral reefs since, what, 2014?
Mm.
And spreading.
That's right, 2014.
Yeah.
And yeah, it's spread way beyond Florida now, across the Caribbean. It affects, I think, over 30 different coral species. It's really devastating.
It's wiping them out. An ecological emergency, really.
It truly is. And the main tool we've had, well, it's basically applying antibiotics directly onto the sick parts of the coral.
Right, I've heard about that.
Mm.
But it sounds like it has limitations.
Oh, big time. I mean,
Like-
think about it. You need divers to apply it lesion by lesion, often repeatedly. It's super labor intensive.
Yeah.
And then there's the worry, about potentially contributing to antibiotic resistance in the ocean. That's always a concern.
So we desperately need better options, more sustainable ones.
Yep.
And that brings us to what we're digging into today, this really interesting idea of using beneficial microbes, like coral probiotics.
That's the concept. The idea is, can we maybe find naturally occurring microbes, perhaps from corals that seem resistant to the disease, and use them to help protect vulnerable corals?
Using good bacteria to fight the bad.
Sort of, yeah. Or maybe just helping the coral's own defenses.
Okay, so for this deep dive, we're really going to unpack a specific study. They took one of these potential probiotics, a strain called Pseudoalteromonas SP strain MH17, out of the lab and tested it right there on the reef.
In situ, as they say.
In the real world.
Yeah. So we'll walk you through how they did it, what actually worked, and spoiler, one method worked really well, what didn't, and kind of what the coral's own microbiome told them about why.
It's a great example of trying to translate that lab potential into something practical against a really tough problem.
Okay, so before we get to the potential fix, let's just quickly paint a clearer picture of the disease itself, SCTLD. Right. So it's this transmissible disease. Key feature is rapid tissue loss, like the coral flesh just slows off, leaving bare skeleton. And it causes high mortality, it kills corals.
And it's a lot of different species, not just one or two.
Over half of all Caribbean reef-building corals are susceptible. It started near Miami in 2014, and now it's confirmed in, I think, at least 30 other locations across the wider Caribbean.
Do we know what actually causes it, like the specific germ?
That's still a million-dollar question, honestly. The exact primary pathogen hasn't been definitively pinned down, but bacteria are really strong suspects. We know this mainly because antibiotics can stop the lesions from progressing, at least temporarily.
Okay.
And researchers often find certain types of bacteria, like various Vibrio species, associated with the disease front. But identifying the main culprit, or maybe it's a group of culprits working together, has been tricky.
And it moves fast between corals. How does it spread?
Yeah, alarmingly fast sometimes. The thinking is it spreads through direct contact sick coral touching a healthy one, and also through the water column.
Like infectious stuff floating in the water.
Could be bacteria, could be infected cells or mucus carried by currents. That makes it really hard to contain.
Which brings us back to why those antibiotic pastes are tough. They only treat the spot you put them on, right? They don't stop a new infection popping up somewhere else on the coral.
Exactly, and you have to keep going back. It's a Band-Aid, a necessary one sometimes, but not a cure-all, and definitely not preventing reinfection or spread.
Hence the need for something different, like these probiotics. So this coral probiotic hypothesis, what's the core idea there?
It's based on the growing understanding that a coral isn't just an animal. It's a host to this incredibly complex community of microbes, bacteria, archaea, fungi, viruses, its microbiome.
Like our gut microbiome.
Precisely. And the hypothesis is that the health of this microbial community is crucial for the coral's health. So maybe we can manipulate it for the better by adding specific beneficial microbes or probiotics.
And what would these good microbes actually do for the coral?
A few things, potentially. They might produce compounds that directly inhibit pathogens, like little chemical weapons.
Okay.
Or they could produce antioxidants to help the coral deal with stress, or maybe even provide essential nutrients, like vitamins. They could also just outcompete the bad guys for space or resources.
So becoming part of the coral's natural defense team.
That's the hope.
And this specific strain they tested, MH17, Pseudoalteromonas species, where did they find that one?
It was isolated from a piece of a great star coral, Montestrella cavernosa, that was collected from a reef in Florida and seemed to be resistant to SCTLD, even though other corals around it were sick.
So they figured maybe it had some good microbes helping it out.
Exactly. And lab work backed that up. MEK-H17 was shown to produce antibacterial compounds, including things called coromycins, which are known to work against a range of bacteria, including Vibrio species often linked to coral diseases.
Okay.
And importantly, in aquarium experiments, applying MEK-H17 slowed or stopped SCTLD lesions in over 60% of the treated coral fragments.
So things looked really promising in the lab. But as you said, the reef is a different beast.
Totally different. Much more complex, uncontrolled conditions. So the goal of this big study was, can MEK-H17 actually work out there? Is it feasible to even apply it effectively?
And where did they set up this field trial?
Off the coast of Fort Lauderdale in southeast Florida, an area, sadly, that's been hit hard by SCTLD.
And they focused on the same coral species it came from, right? Montastrea cavernosa.
Yes, they tagged 40 colonies of Montastrea cavernosa that were already showing signs of active SCTLD, and they monitored them for a long time, 2.5 years, September 2020 to March 2023.
Wow, 2.5 years. That's commitment. And they split these 40 corals into 5 groups.
Correct. 5 different treatments to compare. You had the probiotic bag group,
Okay.
a control bag group, which just got seawater in the bag, then a probiotic paste group, a control paste group using just the paste material without the probiotic, and finally a background control group, corals that were monitored but received no treatment at all, just to see how the disease progressed naturally at that site during that time.
Okay, let's break down those 2 application methods, the bag and the paste. How did the bag work?
So for the bag method, divers would actually enclose the entire coral colony underwater inside a big plastic bag.
A whole thing.
The whole colony. Then they'd inject the probiotic, MH17 suspended in seawater, into the bag. They'd seal it up and leave it there for about 2 hours.
Like giving the coral a full-body probiotic soak.
Pretty much, yeah, a whole colony exposure. Then they'd remove the bag. The control group got the same bag treatment, but just with filtered seawater inside, no probiotic.
Okay. And the paste. That sounds more like the antibiotic approach.
It is more targeted. For this, they mix the probiotic bacteria into a kind of sticky paste made with sodium alginate.
What's sodium alginate?
It's derived from seaweed, often used as a thickener. It creates this gel-like consistency that helps the paste stick to the coral tissue underwater.
Ah.
So they'd use syringes to apply this probiotic paste directly onto the active disease lesion, the edge where the tissue was dying. The control paste was the same alginate base, just no MEK-H17 mixed in.
Got it. So whole colony bath versus targeted wound dressing. And they didn't just treat them once.
No, they applied the treatments multiple times, especially during the first few months, about 4.5 months into the experiment.
And I think you mentioned, or I saw in the notes, they did some safety checks first, making sure this microbe wouldn't harm other corals.
Absolutely essential step. Before the field trial, they did tests in aquaria. They exposed fragments of 5 other common Caribbean coral species to MEK-H17.
Yeah.
And none of those species showed any visible negative reactions, no signs of stress or tissue damage. That gave them confidence it wasn't likely to be broadly harmful to other reef inhabitants.
Good. Okay, so preparing this stuff for the field. How did they get the bacteria ready and make sure it was still, alive and active when it hit the water?
Yeah, logistics are key. They grew MEK-H17 in large batches in the lab. Then they concentrated the bacterial cells.
Right.
This concentrate was then either mixed into filtered seawater right before use for the bagging method, or mixed into that sodium alginate paste vehicle for the paste method.
Could it survive the trip out to the reef, on the boat, etc.?
They checked that too. They tested how well it survived being stored. The good news was it stayed viable alive and potentially active for up to about 2 weeks, even at room temperature, around 22 Celsius.
Oh, wow.
Yeah, and storing it cold, like in a cooler at 4 degrees Celsius, seemed maybe slightly better over longer periods, like weeks, though it wasn't a huge statistical difference over 56 days. But basically, yes, it was stable enough for practical field use. You could prep it and take it out for a treatment trip.
Okay, great. So they've got the probiotic ready, they got their methods, the bags, the pastes, the control groups. They set it all up on the reef. 2.5 years later, what happened? How did they even measure success?
The main way they measured success was by tracking how much living tissue was left on each coral over time. Did the treatment slow down or stop the tissue loss caused by SCTLD?
How do you measure coral tissue accurately underwater?
They used a technique called 3D photogrammetry. Divers take lots and lots of overlapping photos of each coral colony from many different angles.
Right.
Software then stitches all those photos together to create a very detailed, measurable 3D model of the coral.
Wow. Okay.
From these models, they could calculate the surface area of the living tissue very precisely at different time points throughout the 2.5 years. Then they could see the proportional change, how much tissue was lost relative to the start.
So much better than just guessing by eye.
Much, much more accurate for quantifying that loss.
Okay, so what did these 3D models reveal? Which treatment, if any, made a difference?
The results were really striking. And yes, the type of treatment made a huge difference in how much tissue the corals kept. There's a very clear winner and, well, a very clear loser.
Okay, let's hear the good news first. The winner.
The probiotic bag method. This was the big success story.
The whole colony soaked.
Exactly. Those colonies, the ones that got the EK17 bath, experienced significantly less tissue loss over the 2.5 years compared to all the other groups.
Significantly less. How much less?
At the end of the study, the probiotic bag group had, on average, 79% of their initial tissue area still remaining.
79%.
Uh-huh.
That sounds amazing for disease this bad.
It is. It showed that this method really could slow down, or in some cases maybe even stop, the progression of SCTLD in the field over a long period. A huge positive result.
Wow. Okay, so if that was the clear winner, what about the loser? The probiotic paste.
Yeah, this was the really surprising and frankly concerning part of the results. The probiotic paste applied directly to the lesions. Those colonies actually lost more tissue than the corals that got the control the paste with no probiotic.
More. How much more?
The probiotic paste group ended up with only about 27.8% of their tissue remaining. Compare that to the control paste group, which had 59.2% remaining.
Whoa. So putting the probiotic directly on the wound seemed to make it worse.
The data strongly suggests that, yeah, it wasn't just ineffective, it seemed potentially detrimental, maybe exacerbating the tissue loss right at that already stressed disease front. That was completely unexpected.
That is really counterintuitive. What about the other controls? The bag with just seawater and the background ones left alone?
So the background control group, the ones just monitored, they fared pretty poorly, ending up with only about 33.6% tissue left. So the disease was definitely active and progressing at the site.
Okay.
Interestingly, both the control bag and the control paste treatments did better than doing nothing. The control bag had, I think, around 50% tissue left, and the control paste, as I said, nearly 60%.
So even the paste base itself might have offered some slight, maybe physical protection.
That's a possibility. Maybe the alginate paste provided a temporary barrier or interacted with the coral's mucus layer in some way that gave a minor benefit compared to just leaving the lesion exposed. But clearly adding the probiotic to the paste was not the way to go here.
Okay, so summarizing the outcomes. Probiotic bag, big win. Probiotic paste, big loss, potentially harmful. Controls, somewhere in between, but better than nothing.
That's the gist of the tissue loss results, yes.
Now, to understand why, especially why the bag worked so well even if the paste didn't,
Yeah.
they had to look deeper, right, into the microbes themselves.
Exactly. Measuring tissue loss tells you what happened, but the microbiome analysis helps understand how or why. They took samples of coral tissue and mucus before the treatment started, and then again at 2 later time points.
To see if the probiotic actually established itself, or if the treatments changed the overall microbial community.
Precisely. They wanted to characterize the bacteria and archaea living with the corals in each treatment group.
And how did they analyze those samples? What techniques?
They used 2 main approaches. First, 16S RNA gene sequencing. You can think of that as taking a census of the microbial community.
Okay.
It tells you roughly who's there, what types of bacteria and archaea are present, and in what relative proportions. It gives you the overall community structure.
A broad overview. And the second technique?
That was droplet digital PCR, or ddPCR. This is much more targeted and sensitive.
How so?
It allows you to count the exact number of copies of a very specific gene sequence in a sample. So they designed assays to look specifically for a gene unique to their probiotic strain, MPH17.
Like a genetic fingerprint for just that strain.
Exactly. And they also used it to look for a specific gene associated with a suspected SCTLD pathogen, Vibrio corallolyticus. It's like looking for a needle in a haystack with high precision.
Got it. Broad census with 16S, specific counts with ddPCR. So what did they find? Did MPH17 take over the microbiome in those successful bagged corals?
Here's another really key finding, and it matches what they saw in earlier lab studies. No, it didn't.
It didn't, even though the treatment worked.
Correct. The overall microbial community composition wasn't drastically changed by the probiotic treatment itself. The biggest factor influencing the microbiome seemed to be just time, seasonality.
Meaning the microbial communities naturally shifted over the months, regardless of treatment.
Yeah, likely due to changes in temperature, nutrients, or other environmental factors between sampling points they sampled in different seasons. That temporal variation was stronger than the treatment effect on the overall community structure revealed by 16S sequencing.
So the success of the bagging wasn't because NICH17 suddenly became the dominant microbe.
Doesn't look like it. Now, when they looked at the general Pseudoalteromonas group the genus MICH17 belongs to using the 16S data, they did see some seasonal changes. It tended to be more abundant in cooler months, like October and January. Vibrio, another key genus, tended to be higher in the warmer month they sampled, August, but these were general trends across groups.
Okay, but what about the specific ddPCR results for the actual strain they added, MHH17?
That's where it gets nuanced. The ddPCR, looking for that specific MHH17 gene, showed that the probiotic was present in the treated corals, particularly the bagged ones, but generally at low levels.
Low levels.
Yeah. It suggests that MHH17 could persist, it could stick around after the treatment, but it wasn't blooming or taking over to become a huge part of the community biomass. It remained a minor player, quantitatively speaking.
Huh. And what about the pathogen they looked for with ddPCR, Vibrio coralliticus?
That specific virulence gene marker associated with V. coralliticus was also detected, but again, at low levels across all the samples. There weren't significant differences between treatments or over time.
What does that mean?
Well, it could mean a few things. Maybe the Vibrio species present in these corals weren't predominantly the pathogenic V. coralliticus strain carrying that specific gene. Or maybe that gene isn't the sole driver of SCTLD virulence. It adds to the complexity of pinpointing the exact SCTLD pathogen.
Okay, so if the probiotic MHH17 wasn't dominating, even in the successful bag treatment, how does that explain the success? How did the bag method work so well?
That's the really interesting puzzle, right? It suggests the mechanism might be more subtle than just overwhelming the native microbiome.
Like what?
Maybe the whole colony bagging delivered a sufficient initial dose to allow MHH17 to colonize key spots, maybe near the disease lesion or in the mucus layer, even if it didn't grow to high numbers overall.
Okay.
Or perhaps even at low abundance, it was producing enough of those beneficial compounds like the antimicrobials right where they were needed locally to inhibit the pathogen or help the coral's defenses. It might be about targeted action, not necessarily high numbers.
So it's not just about quantity, but maybe what it's doing or where it's doing it.
That seems like a very plausible explanation based on these findings. Quality over quantity, perhaps.
And that could also help explain why the paste failed.
Potentially. Maybe applying it just to the lesion wasn't enough to protect the whole coral from new infection points. Or, as the tissue loss data suggests, maybe that high concentration directly on the already compromised diseased tissue was actively harmful. Perhaps it stressed the cells further, or the paste vehicle itself interfered negatively at that site when combined with the probiotic.
Fascinating. Okay,
Yeah.
let's try and pull the key messages together here. For people trying to manage SCTLD on the reefs, what's the big takeaway?
The biggest, clearest takeaway is that this probiotic strain, MHH17, when delivered using that whole colony bagging method, provided real, significant, and long-lasting protection against SCTLD for Montesoria cavernosa in a field setting. That specific method worked.
And the flip side?
The paste method, applying the same probiotic directly to the lesion, not only didn't work, but seemed to make things worse. So delivery method is absolutely critical here.
Now, practically speaking, bagging a whole coral sounds like a lot more work than just squirting paste. Is that bogging method actually feasible?
It definitely is more effort intensive. You need the bags, the weights, the divers skilled in deploying and retrieving them carefully without damaging the coral. It takes more time underwater per colony compared to paste application. But given how effective it was, 79% tissue remaining after 2.5 years versus maybe 30-something% for untreated corals, the researchers argue the benefit massively outweighs the extra operational costs and effort, especially for high-value or critical coral colonies.
And the probiotic itself is stable enough to get out there.
Yes, the shelf life seems workable for field logistics.
What about wider safety concerns? Putting bacteria into the reef environment.
Well, a few points there. First, it's a native strain isolated from a Florida coral, not something foreign.
Okay.
Second, the initial lab tests on other coral species were encouraging, showing no harm. Third, other research suggests these kinds of probiotic applications tend to primarily affect the host coral's microbiome and don't seem to cause major lasting shifts in the surrounding seawater microbial community.
So limited off-target effects, potentially.
Potentially, but obviously, if you were to scale this up massively, you'd need ongoing monitoring to confirm that. Prudence is always needed.
So it sounds like probiotics, used carefully and with the right delivery method, could be a really valuable addition to the toolbox for fighting SCTLD.
Absolutely. It's not a silver bullet that will solve everything overnight, but it offers a biologically based approach that could complement other strategies, like applying antibiotics in certain situations or restoring corals.
Are there still big questions to answer? Where does the research go next?
Oh, for sure. A lot of questions. Like, for the bagging method, is 1 2-hour application enough for long-term protection, or do you need follow-up treatments? That has big implications for effort.
Right.
Can the delivery method be optimized? Maybe find ways to deliver a whole colony treatment that don't require retrieving a plastic bag, something biodegradable, perhaps, or a different technique altogether.
Sealing it up is key.
Exactly. And fundamentally understanding that mechanism better. How exactly is MIC-H17 helping at those seemingly low concentrations? Is it the cormycins? Is it stimulating the coral immune system? Something else?
Knowing that could help find even better probiotics or methods.
Precisely. And also, thinking about those seasonal shifts they saw in the microbiome, does the timing of treatment matter? Should we treat corals in certain seasons when they might be naturally more vulnerable or when the probiotic might establish better?
Lots to explore still. So recapping this deep dive, specific probiotic, MIC-H17, put in a bag around the whole Montestrella cavernosa coral, gave it significant long-term defense against SCTLD out on the Florida Reef over 2.5 years. The paste didn't work.
That's the core finding. And it's a really hopeful sign in the face of such a destructive disease. Finding new tools, especially ones derived from nature itself, is just crucial if we want to give these reefs a fighting chance.
It really makes you think, about those natural microbial communities already living with the corals, shifting with the seasons, potentially influencing health day to day. It leaves you with a question, doesn't it? What other beneficial partnerships are already out there on the reef between corals and their local microbes that we just haven't fully grasped yet? Partnerships we could potentially learn from, maybe even leverage, to help corals build resilience against all the challenges coming their way.